Hello, and welcome to the CAP Today webinar for Tuesday, December the 5th. I'm Bob McGonigal, the publisher of CAP Today, and I'll be your host and moderate a question and answer session after our formal presentation. Today's webinar is entitled Implementation of an FDA-cleared tumor profiling assay to enable localized precision oncology testing for patients. This very important webinar is made possible by a special educational grant from LabCorp. We want to acknowledge LabCorp and thank them for their essential support that allows us to bring you this important program. We have three speakers today, and I'll say more about each one in due course. All of them are from the University of Alabama at Birmingham. And in the order in which they'll be speaking, they are Dr. Alexander Craig MacKinnon, Jr., MD, PhD, Dr. Shuko Harada, MD, and Dr. Darshan Chandrashekar. He's a PhD and assistant professor at the UAB Birmingham. As I said, I'll say more about them in due course. But before we begin, let me say and share a few housekeeping tips that I've found useful to enhance your enjoyment. First, I often recommend that you refresh your browser. You'll see at the bottom right of your screen an 800 number, 858-345-5916. We have live help available throughout today's webinar. Real people are at the phone and also on the platform to handle any technical questions you might have regarding the website or the audio. You can also, in the question and answer box that you see below the slides, type in any technical problem or question, and that'll be resolved very quickly, again, with live help. Of course, the principal reason that we have a question and answer box is for you to pose your questions and comments to our speakers as they go through the webinar today. At the end of their presentation, as I said, we'll start with those questions and comments and get through as many as we possibly can before the top of the hour's up. I want you to know that we'll have the full audio and slides of today's presentation available at captodayonline.com in about a week or 10 days. You will see some email follow-up from ourselves and possibly LabCorp, so I wanted you to be alert. Finally, it's very important for me to note that CAP Today does not endorse any products or services that may be named in our webinar today. Any remarks of mine are purely personal, not to be taken as the policy of the CAP Today or the College of American Pathologists. And now, let me introduce our first of three speakers. Dr. Alexander Craig MacKinnon, Jr. is an MD and PhD. He's a professor from the Department of Pathology at UAB Birmingham. He trained at the University of Chicago, and as you all know, he's a very distinguished author and lecturer. And I will invite Dr. MacKinnon to begin his part of the presentation now. Dr. MacKinnon.
Thank you, Bob, and welcome everyone. I'd like to start by providing a little background about UAB and our patient population to provide a context for our decision to implement the Ilio-Tissue Complete assay in the UAB labs. So UAB is located in Birmingham, which is in the north central part of the state. It's just up the road from Tuscaloosa, which is where the main Alabama campus is. It's one of two cancer centers in Alabama, and it serves a large catchment area of just over 5 million people when you consider the bordering states. It's the largest employer in the state and has a large economic footprint, over $12 billion in revenue annually. And it was rated #1 by Forbes as the best large employer in 2021, which is something we're all proud of. And the mission at UAB is to improve the healthcare for our state's population. And our group is mainly focused on cancer. And this is a big need and a big challenge in our state. Reasons for this is that a large percent of our population live in rural communities, and over half the population or live in areas that are deemed to be medically underserved, and has a very high overall cancer mortality burden compared to the rest of the country. So this next slide gives you a kind of a high-level view of our test menu. And on the left side is our NGS testing, and it includes on here the Ilio Tissue Complete. But our NGS workflow is hooked up with the hospital EMR and all our analysis software through a LIMS system, which really helps to facilitate our operations. And we've divided our NGS into two main tiers of testing. We have a small panels or hotspots that test less than 50 genes. We run this on the Ion Torrent Genexus platform, and these all have rapid 2-day turnaround times, highly focused, and we run a OPA, which is a solid tumor, and OMA assay. Our more comprehensive genomic profiling, these are like larger than 100 genes. We run these on the Illumina platform. They have a longer turnaround time, about 7 to 10 days. They require higher DNA inputs. So one of the decision factors, whether we go with comprehensive genomic profiling or small hot pot testing, often is determined by the size of the tissue when we first evaluate it. So for the comprehensive profiling, we have two assays, the Ilio Tissue Complete, which we're talking about today, DNA-based assay, it's FDA-cleared, and an Archer fusion panel, which I won't say any more about. Okay, so this graph gives you a look at, prior to implementing Ilio, what our test volumes were like for both in-house testing and send-out testing. And there were two major points here. One is that over the 2 years when we were tracking this data, the testing volume incrementally increased month after month. So it's a growing area of testing, and more cases are coming in. And then we also recognize at UAB, when we were running our hotspot panel, that our test volume kind of saturated. This came, as shown in this graph here, between Q1 and Q2, we implemented the new OPA hotspot panel. And then between Q2 and Q3, we implemented electronic ordering. And after that, the testing volumes kind of leveled off. So this helped us to recognize some of the challenges with our situation and the desire to bring more testing in-house. For one, send-out testing has a laborious, somewhat of a strain on the lab. There's a lot of handoffs and touch points. Many of them require the supervision of a pathologist. Especially with cases that are older and maybe have been filed away in the archives, it can take a long time to review a case, prepare it, and get it ready for send out. So that's this pre-analytical turnaround time that gets extended. And when you compare that to in-house testing, there's much fewer steps involved. It's a lot easier to do. And so there's a real incentive here to find ways to balance the send out and in-house testing so that the patient gets the testing they need, but the lab and the operations can function efficiently and harmoniously with each other. Okay, so given this environment of growth in NGS testing and interest and increase in our in-house testing volumes, we recognize many challenges I think most labs also face. One is developing a competitive test menu. We recognize that the hotspot test menu, although it does serve a very important role, particularly for certain types of cases and very small cases where the tissue is limiting, is not competitive when you need to have larger targets, more targets, and more genetic signatures, which we'll talk about in a minute. And also, there's a shortage of lab professionals and directors and staff to implement these new testing. And one thing we've recognized here, and I'm sure it's common across most of the people who are listening to this, is that it's key to get buy-in from our stakeholders, mainly the oncologists and our cancer centers, so that the testing we develop and offer aligns a lot with their expectations and needs for the care of their patients. You know, and also at a big operation like UAB, you have labs spread out all over campus. The histology can be in one building, the molecular lab in another, cytogenetics in a third, and things get fragmented, and that can lead to redundancies in operations because each lab might have its own LIMS system, or their techs who can do the same job but work in different areas, complicated workflows, challenges with staffing, and then all the informatics and IT and ancillary support you need to provide NGS testing. You need to gather and make sure that it's secure and stable and allows you to function. And then lastly, you need buy-in from the hospital admin who are going to fund your lab, and you need to make sure that it's, there's quality and turnaround time are always an important marker of getting their input, but you also need to grow your testing to keep your operation moving along. So you need a marketing team, billing, customer service, and everything to manage your sales and revenue of your testing. So given these challenges, myself and a student who was in our master's for health administration program conducted a survey of our oncologists to kind of get insight into what were the major drivers that were motivating them to pick labs for sending out for NGS testing for solid tumor. So we sent out a survey, and Dr. Tran compiled all the results, and they're shown here. And basically, each one of these graphs is a different metric we queried about. The more green you see is the more favorable the response. The more blue you see, the least favorable. And they're ranked here in order. And we thought for sure comprehensive genomic profiling would be the main driver that oncologists were using to select testing. We were kind of surprised to find out that even more important than that were the ease of ordering, the turnaround time, and the cost to patients and customer service. And this is really informative to us because from this we recognize that we have control over how orders are processed. We also have control over turnaround time. So these are things that we can improve and help to get more utilization of our lab and be successful with our desire to launch these assays. So putting all this together, we recognize the need for comprehensive genomic testing, mainly to provide tumor mutation burden, our TMB signatures, and MSI testing. We also recognize a great opportunity to consolidate our genetic labs into one physical space and integrate our workflows. This represented a big operational value to the UAB stakeholders, and it also contributed to achieving faster turnaround times. And then in addition to our workflows, there's other things that we are working on to increase our efficiency in our testing, and that's to support clinical consultations by a molecular tumor board, matching patients to clinical trials, which is a major goal of the UAB Cancer Center, and then creating a database that in a vendor agnostic fashion can pull in all the results from the NGS testing into an organized database and then allow for research or cohort discovery or finding samples or things that make going through your records much easier. Okay, so given that, we are in the process of integrating all the genetic labs at UAB that historically had been separate. They lived in different departments or different centers within campus. We're currently doing that now, and hopefully next year or maybe in 2025, our building will be finished and will be all integrated and launched as the Venn lab. And this will help with the volumes and the turnaround times and the clinical expertise that we can provide. So with that, I'd like to kind of switch now and discuss more the ileotissue complete assay and give you a description of the product and some of the features that it has that were really compelling to us when we were making the decision and evaluating assays and picking which one we wanted to go to. Well, one of the most important aspects or features of this assay is that it's an FDA-cleared kit. And given that there's motion by the FDA to regulate lab-developed testing, this seemed like a great way to hedge against that and have a kit that had already been FDA-cleared. It's a clinically focused assay, and it's designed to identify actionable biomarkers in a range of tumor types. And it works with a server that's provided by PGDX or LabCorp. They're related companies. And one advantage of this server is that you need it to analyze the data, but it sits on your local network. And this had some serious advantages for us. One is it avoids issues associated with cloud computing. Different institutions have different policies or opinions regarding the use of cloud computing. Some places are very conservative and make it hard to use cloud computing. So by sticking this onto your own network, you avoid that whole issue. It sits behind the firewall. And second, it also made it easier for here at UAB to have the security review by our hospital information systems to make sure that the server would comply with their whole network and not produce any security risk. And lastly, it's a reliable, efficient 5-day workflow. The overall clinical pass rate, so what this means is what percent of samples that are submitted for the PGDX or the ileotissue complete testing are pass and actually give you a result. And the vendor states at 92.9%. And in our hands, having run the assay for about 9 months now, we're at 91%, so very close to that. It's a DNA-based assay. So your input is DNA, and the DNA is derived from formalin-fixed paraffin-embedded tissue blocks or unstained slides. 100 nanograms of total input DNA is recommended. You can go down as low as 50, and in certain instances, we've pushed it a little further, and you can get results below that, but 50 is a recommended low point. You need at least 20% tumor purity. And when you have adequate specimen, say, for example, 10 slides, and on each slide you have more than 20% purity, you have around a 98% sufficiency rate. And what we found in our hands is we had a 96% DNA sufficiency rate. Very few cases we saw tissue and tested it but not get a result. So very much in line with what the vendor is putting in their advertisements or description of the product. So the assay is mainly looking at SNVs and indels in a large number of genes. They're shown here on this slide on the right side. And in addition to the SV and indels you get across all the exons of these genes, there's two genomic signatures. That's the tumor mutation burden and the microsatellite instability score. There's amplification of the gene ERB-B2, and there's also 4 translocations that are detected, and that's ALK, NTRK2, and NTRK3, and RET. Now, this is what I'd like to point out. This is when you run the assay in the FDA-cleared IVD mode. So it's kind of a locked down version of the assay, and this is the results you get out. There's additional targets, and my colleague, Dr. Harada, will talk about this in much more detail after I'm done. But these are what we call the RUO mode, or the RUO targets. And you can see here, there's a lot more amplifications and important translocations. And what I believe she'll talk about in more detail is the ability to validate these additional targets and include them in your final report when you run this assay. Okay. Another important aspect of this assay that was compelling to us was the fact that CMS has given it a national reimbursement code, or a PLA code that works across all 7 MACs. So it's approved for Medicare patients and these MACs, and the reimbursement rate allows you to run this test and generate a slight margin that is beneficial for the financial health of your lab. At UAB, we also got a Z code, which is additional information for the test for billing purposes. Now, in terms of commercial payers and what their approach to this assay is, it's kind of a mixed bag, and it really heavily depends on what part of the country you're in and who the providers are in your area. Some providers will cover this, other ones won't, and that's just something that when you go to set up this assay, you'll need to find out based upon your geographical region and where you're working. Okay, so the test results itself come out in a nice format. It's very succinct. The first is a high-level summary of the test. It gives you the disease at the patient had, some of the sequencing metrics, number of mutations. But then, the results are broken down into 2 major categories. You have variants with evidence of clinical significance, and very similarly, variants with potential clinical significance. So what does this mean? Okay, so variants with evidence of clinical significance. This more or less correlates with the AMP-ASCO-CAP NGS reporting guidelines that are known as Tier 1. And kind of another way of thinking about it, these are all targets for which there's an FDA-approved therapy for that tumor type. And the variants that are listed under the category potential clinical significance, these align with Tier 1B through Tier 4 evidence, in which there's strong supporting evidence that there's clinical relevance to them, and they're included in the second section of the report. So another way of kind of conceptualizing this is the AMP tiering system that was proposed and used by many labs, and the variants with evidence of clinical significance are in this level A, this Tier 1A, and the variants with potential clinical significance are in 1B and the other levels too, and they have stronger evidence but not FDA clearance. Okay, what we do at our institution is, in addition to distributing the ILIO report, we pull all the data from the sequencing and push it into one of our vendor partner, Genome Oncology, and generate a UAB report. And our report is organized based on the pathogenicity of the variants. So it'll give you details on variants that are pathogenic, potentially pathogenic, and and drug response and clinical trials matching and additional information. We bundle the two reports together and distribute them. So this diagram here shows how we have set up the Elio NGS in our so-called molecular ecosystem. So in the top left would be where the NGS data is generated. And we're setting up in a way that it's really vendor agnostic, whether we run a case in our own hospital lab or as a send out case to another national provider lab. The files come back and they're in these different formats like VCF and BAM, which many people are very familiar with. We pull them in through a series of interfaces into our Precision Oncology platform. And this is allows us to structure our NGS data in a manner in which we can integrate it with clinical knowledge. So this could be, by clinical knowledge, what that means is actionability about a certain variant. It could be a clinical trial that exists with that variant being identified, or even additional information from the EMR. And by integrating it in this behind-the-scenes platform, you can then have various user interfaces that will generate your reports, which is shown in the bottom right. It'll be a database that you can mine to find interesting cases or to look at operational efficiencies or whatever interest you may have. It's used to feed into a molecular tumor board, so you can load in your clinical trials you have open at your institution and cross-match them. And then you can run your molecular tumor boards off of this. And by setting up in a vendor-agnostic fashion, it really doesn't matter where the patient is tested. It allows you to pull it all in and hopefully give the patient the best care possible. Okay, so the last couple of slides give you a summary of the verification process. So since this is an FDA-cleared assay, you don't need to go through the extensive validation of a lab-developed test that you're probably more familiar with. This one is a verification. We used 20 cases that we had tested previously on different platforms. There were 40 variants that were predicted to be identified by both panels. It's all summarized in this table. And when we ran them, the number of replicates we had to run and the reproducibility and different texts running it and everything, it had excellent concordance with our pre-existing assays. So Elio Tissue Complete identified 40 of 40 variants that UAB had picked up. And there was one variant that the Elio Tissue Complete assay identified that we had missed, and that was a P53 variant. So we weren't sure what was going on with that. We went and looked up the case, and the variant was in our BAM files. We just aren't sure why it got filtered out, and and for some reason it ended up not getting reported. But anyway, it did very well. It has very high concordance and it performs really well in our hands. So lastly, for my part, we've made this electronic orderable, so the clinicians, if they want to order testing, we have it right up at the same place where the outside tests are, so it's conveniently located, and they can just check the box and initiate the ordering process. So with that, I'd like to thank you very much for your attending today's seminar, and I'm grateful to both CAP Today and the PGDX team for allowing us the opportunity to present today. So I'll hand it back over to Bob.
Dr. MacKinnon, thank you very much. What an interesting presentation. You've generated lots of comments and questions that we'll get to in due course. But do let me remind all of you who are participating today that at any time you can make comments and ask questions in the Q&A box that you see below the final slide. And we're getting a lot of questions, of course, about software systems and ordering systems. A very interesting example there that you finished with. Thank you, Dr. MacKinnon. And now it's my pleasure to introduce Dr. Shuko Harada. Dr. Harada is a professor in the Department of Pathology, Genomic Diagnostics, and Bioinformatics, again at the University of Alabama at Birmingham. She did her residency at Drexel and then did subsequent work at the University of Pennsylvania and Johns Hopkins. It's my great pleasure to introduce her now, and Dr. Harada, you may begin your presentation now. Thank you.
Thank you, Bob. Thank you, everybody, for attending this webinar today. And then I'm going to discuss about our experience on targets beyond IVD, Elio Tissue Complete. So as Dr. MacKinnon illustrated today earlier, the IVD target, especially for the amplification and translocation, is a little limitation, has a little limitation on this assay. It only detects ERBB2, HER2 amplification, and the translocation target is also only ALK, NTRK2, NTRK3, and RET. However, actually, in RUO mode, there are many more targets you can capture with this kit. So we have some experience on our first example is the 66-year-old female with right brain mass. Neuropath diagnosis was glioblastoma, IDH wild type, and CNS WHO grade 4. So the neuropathologist ordered idiotissue complete, a 500 gene panel, and the fusion panel by Archer Fusion Plex. So when we had the results, idiotissue complete detected some pathogenic variants, including EGF receptor, extracellular domain missense mutation, S645C, and TERT promoter mutation. But we're surprised that we saw the exon 2 to 7 skipping by fusion panel detected by using Archer Fusion Plex, which is showing here in the bottom. And this is results in truncation of the EGFR extracellular domain, and also known as EGFR V3, which is seen quite often in the neuro brain tumors. So we went look at the RUO component. There's a RUO complete case record, also produced by the idiotissue complete PGGX pipeline. And if you can see in the bottom, EGFR gene fusion is detected. It's exactly the same as the V3 variant that Archer assay detected. In addition, there's a copy number change event is detected. Its raw fold change is about 20-fold increase, which suggests amplification in this case. That happens quite often together with EGFR exon skipping in a brain tumor. So after this, we had several cases, additional cases experienced, and then most of the case Archer picked up exon skipping, and all of the case had also RUO component has the EGFR exon skipping event code. So this is pretty, we are very happy, confident, be able to pick up by EDO assay. And other RUO gene targets, and a lot of amplifications you can detect. One of them we focused on MDM2. MDM2 gene, as you know, amplification happens in well-differentiated liposarcoma or atypical lipomatous tumor, or de-differentiated liposarcoma derived from well-differentiated liposarcoma. So pathologists order a lot of MDM2 fish to distinguish those entities from the benign lipoma. And FISH is done detecting the MDM2 gene on the chromosome 12 and giving a ratio, we report as a ratio to be amplified or not. So we tested about 10 cases. 5 of them are amplified and 5 of them are non-amplified by FISH using as a gold standard. If you see the second column and third column, MDM2 copy number per cell and MDM2 to chromosome 12 ratio, top 5 showing in red has high high number, and then bottom 6 is less than 2 in ratio, so it's non-amplified. So the EDO, we can check the raw fold change in RUO mode. One case failed by QC, but all the cases that was amplified by FISH has a very high raw fold change, whereas the non-amplified case, everything was less than 2. Raw fold raw fold change is less than 2.5. So if you plotted these all cases in here, it's if we use the cutoff same as the IVD ERBB2 CNV criteria, which is a fold change 2.5 in greater than 25% of the regions covered, then we can perfectly separate non-amplified case, which is shown in blue on the left bottom side, and to the amplified case shown in red. So we could, it is very also happy with this performance that we can trust amplification evaluation. Last one is the beyond RUO, which is interesting, we thought. The patient is 61-year-old case, 61-year-old female with abdominal mass. Pathology diagnosis was gastrointestinal stroma tumor, large size, but low-grade spindle cell type. Pathologic stage was PT3, and then 500 gene panel was ordered, and subsequently oncologist also ordered carries the send-out test. This is brief showing morphology, low power and high power, show spindle cell morphology, and definitely KIT and DOG1 IHC positivity. So PGDX EDO tissue complete results. So here, microsatellite stable tumor mutation burden low, and then didn't find any clinical significant variants. Well, there are 3 potential significant variants are there, but then there's no KIT or PDGF receptor mutations identified. In contrast, when the Caris results came back, we're surprised. They detected exon 11 in frame deletion. So we went back to EDO results, the BAM file, visualizing using IGV, Integrative Genome Viewer. And you see this here on the, let's see, here is the exon 11 of the KIT is showing here, and then this line blanking part is showing large deletion in exon 11, which is same as the one Caris reported, the one from exon codon 560 varying to the tyrosine at codon 578 is deleted, which is a 57 base deletion, quite a large deletion, but certainly it's there. But then we are asking discussing with PDGX, they have a pipeline set up to upper limit of the insertion deletion detection to the 42 base. So anything larger than that is not detected because it's out of the limit. This was apparently based because of the accommodate FDA approval, the size cutoff, they have to put it up. So we asked the bioinformatics team to run on our secondary pipeline analysis, and MUTEC2 and Pindels both picked up the this KIT deletion, 57 base deletion. So this extra pipeline analysis can be implemented for the subsequent analysis of the to detect large deletions insertions, especially for the GIST. So with all this experience, I show here summarizing. We explore the more data beyond IVD portion of this PDGF tissue complete. Show the fusion events and copy number variation. You can use this detect by using RUO mode, and the large insertion deletion detected by our own bioinformatics tool. So more potential in the future, the kind of wish list is homologous recombination deficiency or HRD score detection. Also, they wanted to see copy number loss of certain genes. And with this, these all work cannot be done just by us and everybody. I really thank you, our team effort for all the molecular pathologists, bioinformatics team, molecular lab technologists, and PDGX technical support as well. And I'll send back to microphone to the Bob. Thank you.
Dr. Harada, thank you very much. That was a wonderful tour of the RUO application, which is, of course, extraordinarily important in this area. We really appreciate that presentation. Let me remind all of you, you can make any questions or comments that you'd like to, and we'll deal with those as best we can as we approach the top of the hour. But before our session, we have our third and final speaker. It's my pleasure now to introduce him. And this is Dr. Darshan Chandrashekar. He's a PhD. He is an assistant professor in the Department of Pathology, Genomic Diagnostics, and Bioinformatics, again at the University of Alabama at Birmingham. He trained in India at the Manipal University in Karnataka, and it's a great pleasure to invite him to present the last and final segment of our formal presentation today. Dr. Chandrashekar.
Thank you, Bob. Hi, everyone. Today, I'm going to present an interesting research project from our division where we have used PGDX elio tissue complete assay for the molecular characterization of endometrial carcinoma based on MSI status. MSI, microsatellite instability, is a genetic condition that leads to impaired DNA mismatch repair. About 20 to 40% of endometrial cancer patients are known to show MSI, and patients with high MSI are known to show positive response to immunotherapy compared to fluorouracil-based chemotherapy. Thus, finding about MSI status is critical for clinicians or oncologists to determine the personalized therapy for the patients. However, detection of MSI status in endometrial cancer is challenging due to high incidence of weak MSI phenotype. We began this project with an objective of studying the mutation profile of microsatellite stable and microsatellite unstable endometrial cancer using FDA-approved genomic tests. At the first phase of our project, we actually collected samples from 16 endometrial cancer patients, and these samples were used for two different assays. Immunohistochemistry was firstly performed to identify mismatch repair status, which is equivalent to MSI status. We also did use the same samples for the elio tissue complete assay. And once the sequencing is and analysis is completed in PGDX server, we actually downloaded RUO data, and these data were summarized and visualized using open source R package called MapTools. We analyzed the microsatellite instability status, tumor mutation burden, and mutation profile using the RUO results. Firstly, the immunohistochemistry analysis revealed that out of 16 samples, 4 belong to proficient MMR status, which is equivalent to MSS, and 12 correspond to deficient MMR status, which is equivalent to MSI. This busy slide that you can see shows the OncoPrint results of the entire analysis. The top part of the plot here provides information about the MSI status, TMB state from the IHC, TMB status from the elio assay, and the histology for all the 16 samples. The main part of this particular figure here provides the mutational profile of top 25 most commonly mutated genes across the samples. As you can see, each tile here is colored differently based on the type of mutation, except for the green color tile, which actually indicates multiple mutations were detected for a particular gene in the same sample. So from this plot, one can see that P10, ARID1A, and PIK3CA were commonly mutated across the sample irrespective of MSI status. However, we observed 2 genes. One is ARID1B, which is part of a Swiss-Smith complex involved in gene regulation, and RNF43, which is a ring finger protein, which is involved in ubiquitin transfer function, and this is also a negative regulator of Wnt signaling pathway. Now, these 2 genes were exclusively mutated in MSI tumors, and that's our initial observation from this analysis. So what we did next is we explored these mutations where in terms of their location in the protein sequence and also their recurrence. So we observed that one of the frame shift mutations that we observed in RNF43 are recurrent in 5 different MSI samples. In addition to this, we also looked at the publicly available data to validate our observations. So we took all top 25 mutated genes and looked at the mutation frequency of these genes in a publicly available whole exome data from MSKCC. And here we observed that ARID1B and RNF43 were mutated in 37 and 42% of the total sample from the cohort. In addition to the mutation analysis, we also use the RUO results from PGDx elio tissue complete assay to study the gene amplification across the samples. We didn't find much of interesting results from this, but we did observe the PIK3R1 is amplified in 2 of the MSI samples. In conclusion, the comprehensive analysis of RUO results from PGDx elio tissue complete assay led to the identification of P10, ARID1A, and PIK3 as the topmost mutated genes across the 16 endometrial carcinoma patients, irrespective of the MSI status. And we observed that ARID1B and RNF43 are exclusively mutated in microsatellite unstable endometrial carcinoma. We did see the PIK3R1 amplification in 2 of the MSI endometrial carcinomas. We are in the process of expanding this research project by inclusion of more samples, both MSI and MSS endometrial carcinoma samples. And in future, we are planning to build a multi-component classifier to differentiate MSI endometrial carcinoma from MSS. So with this, I would like to conclude my presentation and thank my colleagues at our division who have helped in the analysis of this particular project. Thank you.
Dr. Chandrashekar, thank you so much. All 3 of you have delivered a magnificent overview of developments at UAB Birmingham, and you're all to be congratulated. We have a good number of questions and comments, and let me go through some of those now. Let me start with you, Dr. MacKinnon. How old have you found that the FFPE blocks can be and still be viable for use with this technology?
That's a good question. We didn't specifically look at dates to do that experiment to know if there's a cutoff. In our validation, we went back several years, so we never ran into an issue with the age of the block, and I would just say probably went back up to 10 years, maybe not quite that far, but that might be somewhat realistic. I would point out, though, that we did see an age of block effect with the Archer assay for some of the targets. They drop off after about 2 years of age.
Very good. Thank you. And while I have you on, Dr. MacKinnon, we've got several questions. You're now going to have a Venn lab, and I'm assuming that has something that's related to the Venn of the Venn diagram. Am I not correct?
Right. It represents the different stakeholders coming together to form the lab.
Yes, I understand. And so what limb system are you considering when you finally bring all that combination together into Venn Lab? Will there be a single limbs or will there be several?
There will be single limbs mainly used for tracking specimens throughout the testing odyssey. We're evaluating them now. We don't have a lead candidate yet. Very open to suggestions if anybody knows of a good one, but it's an active process right now, trying to find that limb system.
I understand. Thank you very much. Dr. Harada, I have a question for you, and let me read it for you. Do you have any concern in using an in-house platform such as PGDx Alio in that it might reduce your ability to identify or resolve certain quality issues such as contamination, sequencing artifacts, sample swaps? I think you get the gist of the question. So I think they want to talk a little bit about the metrics built into the system. Dr. Harada.
Yes, so I don't know why the person thinks reducing ability to detect core issues. I think we have been running NGS assays past 8 years or so, and have, I think, all the quality tracking as well as the sample tracking, the same practice the lab has been doing, and there's no any difference between any different NGS assays. But the good thing on PGDx assays, they analyze all the quality metrics. Darshan may have more details, QC parameters about that. But anyway, so with all this, they pass QC and pass or fail QCs readily. So any poor samples, some reason, it fails QC, so then we cannot report, either repeat the test or the report as a quantity not sufficient, quality not sufficient. So that QC system parameter, along with a positive external control, would definitely give us confidence in assay performance. It's pretty good. I don't know if I answered all the questions, but that's how we monitor the assay.
Thank you. Thank you very much. Dr. MacKinnon, do you have anything to add to that?
No, it's FDA cleared, and so as Dr. Harada was saying, there's sequencing metrics and performance metrics that are built in. So a sample on a run, I think you can load 15 samples in a control in one run. And so an individual sample can fail if it doesn't meet certain metrics, and the whole run can fail if the positive control doesn't identify the expected variance. So I think those are two aspects of the assay that work really well for quality control. And then you have all your pre-analytical control where you examine the tissue and make sure you sample the appropriate areas. And when you combine both of those, I think the performance of this assay is in line with any lab-developed test that you would normally run, where you've gone through the precautions and the ensuring that you know the performance of your assay.
Thank you. Thank you very much. Dr. Chandrashekar, there's a question here to talk about your validation plans for cases that you use for RUO targets. Could you talk a little bit more about what you'd regard as a robust validation plan in the RUO space?
Yeah. So in case of the results that we have, there are two ways. One is the in silico validation, which we generally do. We look into the specific mutation using IGV, as well as we look at the publicly available data where we can observe the similar results for our validation, and eventually our aim is basically to establish the profile. So for MSI and MSS tumors, we want to have enough number of samples. We want to we want to expand this project with more samples on, and then eventually build a classifier. And that classifier would definitely should help in defining or distinguishing the MSI endometrial carcinoma samples from MSS samples based on the molecular signature itself.
Thank you. Thank you very much. Let me ask you, or perhaps I should direct this to Dr. MacKinnon, about how many samples can you test per month using the ELIO and other CGP assays?
Using ELIO, we're running one run a week at present. You probably, if you had 2 shifts in your lab and a large enough demand and volume, you could probably do 2. You're limited by your sequencing. It takes about 2 days, and then your analysis. So we're running, I'd say 15. That'd be 15 patient samples per run. We're running one run per week.
Very good. Thank you. And now the following question is a very good one, and I want to make sure we have adequate time. I'm going to ask Dr. Harada to respond first. Why do you use the Archer fusion panel instead of validating the RUO fusions on the BGDX panel?
Well, yeah, so we wanted to launch PGDX panel quick as possible, so we first went FDA-approved IVD version, and we already had Archer Fusion Plex validated in a few years ago. We have been running this assay. So therefore, as neuropathologists knows that we would miss EGFR, in this case, EGFR exon skipping by PGDX before validating RUO components. Therefore, they order both a fusion panel and a 500 gene panel. Our plan is, of course, to validate that component in RUO component completely, to implement everything, and then report those variants as well in one assay. And also we can report amplification as well.
Thank you very much. Dr. MacKinnon, would you care to make a comment on that same question?
Yeah, that's a great question, and I think I agree with what Shuko said, that we already had the Archer assay existing that works really well, and it was just a kind of a timing to get the ELIO assay up and launched as soon as possible. Since it's a DNA-based assay, it's not as good as maybe an RNA-based fusion assay could be. It does target some of the important ones. So I think that you just have to kind of evaluate for the different diseases, the spectrum of fusions you need to be able to identify. And so that would be a big driver of the decision making.
Okay, thank you. And let me ask as another question for Dr. Harada, the inevitable question, that we always seem to get at some point in a webinar on this subject matter. Have you considered using circulating tumor DNA samples or other blood-based diagnostics with this approach and with this platform? Dr. Harada.
Yeah, so yes, PGDX has a circulating tumor DNA assays as well, in same using a similar kit. We have been talking, and this is our wish list as well. We haven't we haven't made a decision how many genes we wanted to test for this circulating tumor DNA. We wanted to go smaller panel, like focused panel, like 40, 50 genes, versus we want to go 500 genes. The clinician likes the larger the better, but then of course you need a larger sequencing capacity to capture enough depth to be a good sensitivity. So that's decision-making point. We have not made it yet. Yes, but it's definitely on our list.
Very good, thank you. And let me just ask one final question of Dr. MacKinnon. Could you just tell us briefly what has been the reception from your oncologists and other physicians treating patients at UAB so far?
The oncologists at UAB have been very positive about the Ilio-Tissue Complete. A lot of them are happy it's here. They support the in-house testing and the turnaround time. So it's been very positive. Right now we're working on trying to find plans for different disease groups to say what testing should be done in-house, and when it's done in-house, Ilio versus a small hotspot panel, and what tests need to be sent out. And I think having a clarity on that will really help a lot for being efficient and making sure that samples get tested as fast as possible.
Very good. With that, I'm going to give you the last word because we're right up against the top of the hour. This has really been a wonderful presentation. Let me remind you all that in about a week we'll be able to post all the slides and the audio of this great webinar at captoday.online.com. You can review it, you can share it with colleagues, and I'm sure you'll find it very enriching. It's been terrific. So let me begin by thanking Dr. Alexander MacKinnon, Dr. Shuko Harada, and Dr. Darshan Chandrashekar, our three speakers, all from UAB. They were absolutely wonderful. Let me also thank LabCorp for their essential sponsorship through their special educational grant for today. And finally, I want to thank all of you who've taken time to join us today on this CAP Today webinar. We're very aware how busy you are, and so we're always gratified and hope to earn your time with these sorts of presentations. We're very grateful to you. So once more, thanks to Drs. MacKinnon, Harada, Chandrashekar, LabCorp, and all of you. And with that, I'm going to bring today's webinar to a close.